first question, which is a relatively easy question, which is what is the purest type of reagent water? If you've read the book or if you've read Bishop, alam nyo na meron three types of reagent water. Type 1, type 2, and type 3. Type 1 is the purest form, while type 3 is the least pure. So type 3 reagent water is actually also known as autoclave wash water. So as you all know, yung purest form is yung type 1. So type 1 yung sagot kasi siya yung ginagamit for test methods requiring minimum interference. Now, if you want to know more, meron akong mga rationale na linagay sa handouts nyo which delves deeper. Pero it's also important to listen to this video or watch this video for reinforcement. So yun ang answer natin for question number 1. For question number 2, ito naman yung mga type ng questions na nagre-require talaga ng luck, I think. Kasi yung question is, which of the following drinks does not contain alcohol? And maraming nalito dito, and thankfully, as a person na umiinom ng root beer, I know that it is a non-alcoholic drink. Pero mamalasan ka talaga, guys, if hindi ka umiinom ng root beer and you assume na alcoholic siya because of the word beer. Maraming nalito dito, pero ito nga yung mga questions na nagre-require lang talaga ng luck. Finally, what is the angle of the syringe when performing phlebotomy? The answer is 15 to 30 degrees. Any value na within that range could also be given, so try to think more rationally regarding that, guys. So 15 to 30 degrees, ilang beses nang tinanong sa atin to. Syringe when alcoholic siya because of the word beer. Maraming nalito dito, pero ito nga yung mga questions na nagre-require lang talaga ng luck. Finally, what is the angle of the syringe when performing phlebotomy? The answer is 15 to 30 degrees. Any value na within that range could also be given, so try to think more rationally regarding that, guys. So 15 to 30 degrees, ilang beses nang tinanong sa atin to. So, mag-move on na tayo sa next question. So, yung next question natin, guys, nagre-require na memorize natin yung mga formula na feeling natin hindi importante. If I'm not mistaken, sa board exam ko, lumitaw yung mga formula ng molarity, normality. Unfortunately, I cannot remember the exact question na lumitaw. Pero I do remember na merong question doon na pinapa-convert yung Fahrenheit to Celsius. So, ang question dito is convert na 99.2 degrees Fahrenheit to Celsius. So, in your handouts, I've given there sa may bandang rational yung paano i-convert yung Fahrenheit to Celsius and kung paano na rin i-convert yung Celsius to Fahrenheit. Now, yung formula on converting Fahrenheit to Celsius is so 99.2 minus 32. Tapos lahat ng to, if minumultiply natin sa 5 over 9, and yung makukuha nating sagot is 37.33. So, yun yung answer, guys. So, yung next na tanong is, where does the TCA cycle occur? Una, confused ako bakit to lumitaw in the boards. Pero, alam naman natin na yung TCA cycle or yung Krebs cycle, it is something that takes place in the mitochondria, and it is the release of energy stored in the nutrients through oxidation. Of acetyl-CoA. So, where is bile stored in the body? So, yung bile, nagpanic ako sa tanong na to kasi in-overthink ko, pero thankfully in-answer ko yung gallbladder, which is the right answer. And next question is, at what serum concentration does glucose start to become detectable in the urine? So, the answer here is 160 to 180 milligrams per deciliter. Any number na within this range, I'm pretty sure nung boards namin, this was the first question, first exam, first day. And ang lumitaw lang is, I believe, 170 or 180, if I'm not mistaken. Pero any number within that range is the correct answer. So, wag niyong kakalimutan yan, guys. And inuulit-ulit din naman nila sa atin yun. Pioneer or sa review center natin. So, yung next question natin is another easy question, pero siguradong marami talagang malilito. Which of the following is considered a lipid? So, the answer here is cholesterol. Tandaan nyo that chylomicrons, LDL, and HDL are not lipids per se, they are actually lipoproteins. which are lipid carriers. Pero since nga lipoproteins, they are considered as proteins. Aside from cholesterol, we also have triglycerides and fatty acids. They are the ones that we actually consider as lipids. Next question, which lipoprotein is the smallest and is composed of 50% protein? So sa first part pa lang ng question, which lipoprotein is the smallest, makukuha na natin yung sagot. Since smallest siya, we could also assume that it is the densest. And it is composed of 50% protein. The answer is HDL. So sa rationale niya, I listed some of the diameters, the density, and the average compositions in the different classes or different types of lipoproteins. Which of the following would be the most adversely affected by a sample from a non-fasting patient? So we all know that HDL, cholesterol, and LDL do not necessarily require a fasting specimen. Triglyceride actually is the one that is most affected because it is the one that is associated with a milky appearance of serum. Milky serum is the one that is associated with triglycerides. So, it is quite crucial na fasting ang ating patient sample. So, which apoprotein is responsible for receptor binding in IDL and chylomicron remnants? Remember to review the endogenous and exogenous pathway of fat transport. So, chylomicron remnants. Remember to review the endogenous and exogenous pathway of fat transport. So, chylomicron remnants, they are taken up by the liver through ApoE and LDL receptor, and another receptor on the surface of the liver cells called LDL receptor protein. Pero, along with chylomicron remnants, IDL is transported and taken up by the liver via ApoE also. So, both of them are taken up by the liver by ApoE and an LDL receptor, an LDL-related receptor. This explanation is lifted directly from Bishop. So if you have time, please do try to read Bishop. So yung next question natin is, which lipoprotein migrates furthest from the anode in electrophoresis? So hanggang ngayon guys, nalilito pa rin ako dito. Remember that anode is positively charged. It attracts a negatively charged anion. So, sa pole na to, kunwari, positive, ito nun yung anode. I know it's like, dapat alam ko na to by now, pero nalilito pa rin talaga ako. And, so, given na yung two types of tests for separation of lipoproteins, iba yung results nila. Try to memorize yung difference ng results nila or yung exact results for each. So, sa ultracentrifugation, it is more dependent on the lipoprotein's density. Meanwhile, sa electrophoresis naman, it is their mobility in electrophoresis. So, if furthest from the anode, yung pinag-uusapan natin, then, yung nagmamigrate furthest from the anode is yung chylomicrons kasi they remain at the origin, or sila yung pinaka-close sa cathode. So, yung mga terms such as beta-lipoprotein, alpha-lipoprotein, they are actually termed depende kung saan sila nagmamigrate sa electrophoresis. So, yun yung answer natin dito. The answer is chylomicrons. Next question. Which sample is best used for lipoprotein assay and preservation? So I tried to look for the exact source for this, and it's also actually in Bishop. The best sample, according to Bishop, for a lipoprotein assay is actually serum, and ito rin yung ginagamit natin in the laboratory. We use serum to assay for lipoprotein. However, traditionally, EDTA was used kasi its ability to chelate metal ions was thought to promote stability. So the best answer here is EDTA. Which tumor marker is best used to monitor the recurrence of breast cancer? So another thing na makukuha mo lang din sa memorization are the tumor markers. So for breast cancer, the answer is CA15-3. I've listed in your rationale or in your handout the different tumor markers and yung related na cancer sa kanila. Remember, the tumor markers are used to monitor, usually, the progress of cancer within. So yung next question natin is a question na lifted directly from Elsevier. One of my biggest regrets din ng board exam, guys, is hindi ako nagbasa ng Elsevier. And unfortunately, maraming lumabas na hindi lang sa CC, pero sa mga ibang subjects din that you will see later. Nagbasa ako ng Har and ng Shula and konting Turgon, pero hindi rin ako nagbasa ng Elsevier, one of my regrets. So, word per word siya, the question is, a patient sample was assayed, yielding a serum triglyceride value of 1,036 milligrams per deciliter. This caused immediate concern as this triglyceride value is associated with what disease? The answer is pancreatitis or acute pancreatitis. This question is lifted directly from Elsevier, as I said. So, basahin niyo yun guys kasi sayang yung mga ibang points. I think nasagot ko naman 'to ng tama, pero siyempre mas magandang sure pa rin. Next question. What West Guard rule is considered to be the warning rule wherein you do not reject the run? Alam na natin 'to. 1-2S. Kapag 1-2S, hindi mo pa i-reject yung run. Instead, this is a warning rule. I also noted na kailangan alam niyo yung itsura kapag nakalagay na siya sa LJ chart or sa Levy-Jennings chart. Anong itsura ng 1-2S? Like, hindi pwedeng alam niyo lang yung definition niya kasi may chance na baka yun yung lumitaw. You'll see later bakit ko iniisip yun. So, ayun yung mga different procedures. I-memorize nyo din kung alin yung mga systematic error, ano yung mga random error. May chance din na yun yung lumitaw sa boards nyo. Next question. Kaya ko sinasabi na kailangan alam nyo dapat yung itsura is because ito yung lumitaw nung boards namin. So, identify the type of error seen on days 10 to 16 of this chart. So, ito yung chart or like, ganito yung chart na lumitaw. So, tingnan mo yung days 10 to 16. Obviously, merong clear downward trend. So, trend yung sagot. Dapat alam niyo yung itsura ng trend, guys. Alam niyo yung either increase or decrease for 6 consecutive days. So, 10 to 16, 6 days yon. Meron din dyan yung definitions ng iba yung trend, shift, outliers. Alam nyo dapat yung itsura ng mga yan. I suggest na i-review nyo na yung mga pictures ng LG chart niyan. Next question. How will you interpret the following results? Control area is negative, test area is positive. Another common sense question. Syempre, if yung control mo is negative, it should always be positive. If control mo is negative, the result is invalid. Like example sa mga pregnancy test, syempre kailangan merong isang line. If walang line at all, then hindi pa rin yun considered as a negative result. Invalid yung result na yun. Next is Deficiency of hypoxanthine guanine phosphoribosyltransferase leads to what disease state? So the answer here is Lex-Nyhan syndrome. So ito yung merong orange sands sa diaper. Orange sands. Due to the presence of what? Due to the presence of uric acid. So itong enzyme na to is involved in the metabolism of uric acid in the body. So kapag multiple myeloma, usually yung associated natin dito is yung plasma cells or yung Bence-Jones in sa protein. Sa Hurler syndrome, this is an example of mucopolysaccharidosis na merong lysosomal deficiency or lysosomal enzyme deficiency rather. And Hartnup disease naman is yung deficiency sa amino acid tryptophan, and this causes blue diaper syndrome. So, isa rin to sa mga techniques ko, guys. Lysosomal na merong lysosomal deficiency. Sa Hurler syndrome, this is an example of mucopolysaccharidosis na merong lysosomal deficiency or lysosomal enzyme deficiency rather. And Hartnup disease naman is yung deficiency sa amino acid tryptophan, and this causes blue diaper syndrome. So, isa rin to sa mga techniques ko, guys, when I was reviewing for the last few weeks or maybe last week before my boards. Kumukuha ako ng mga random na practice test and rinarationalize ko, pati yung mga ibang choices na mali, iniisip ko, bakit sila mali? Ganun. And I think na very nice way yun to review, to recall, para talagang magtatak sa'yo yung mga questions. Hindi mo lang dapat alam yung tama, alam mo bakit mali yung ibang choices. Siyempre, I recommend this if marami pa kayong time na mag-review, guys. Pero sa mga last few hours or last few days, try mo na lang ipasok yung kaya mong ipasok. Last few hours or last few days, try mo na lang ipasok yung kaya mong ipasok. So, how is tolbutamide administered in tolbutamide testing? So, yung tolbutamide tolerance test is done by dissolving 1 gram of sodium tolbutamide in 10 ml of sterile water. It is administered intravenously, yun na yun, over a 2-minute period. So, itong exact na rational na to or exact statement na to is galing sa mismong journal na nagde-describe ng tolbutamide tolerance test. So the answer is intravenously. What is the major analyte that affects osmolality? major analyte that affects osmolality. So, thankfully, hindi lumitaw yung mga ibang analytes na nag-a-affect ng osmolality. Pero if alam natin naman is yung major na nag-a-affect, that is sodium. So, on to the next question. Na nag-a-affect, that is sodium. So, on to the next question. Yung next question natin is, what is the most affected analyte in prolonged tourniquet application? So, you know what happens in prolonged tourniquet application is that there is hemoconcentration and hemolysis. So, if there is hemolysis, what happens is yung cellular content ng RBC, which is yung intracellular cation, which is potassium, it will be present in the plasma or the serum. So, kapag nagkaroon ng prolonged tourniquet application, yung most affected analyte is potassium. It will be present in the plasma or the serum. So, kapag nagkaroon ng prolonged tourniquet application, yung most affected analyte is potassium. Is yung cellular content ng RBC, which is yung intracellular cation, which is potassium, it will be present in the plasma or the serum. So, kapag nagkaroon ng prolonged tourniquet application, yung most affected analyte is potassium. Which process involves the breakdown of pyruvate to lactate? Keyword natin dito is breakdown. So, yung choices natin are mga processes na related sa glucose metabolism. There's gluconeogenesis, which is yung formation of glucose from non-carbohydrate carbon substrates. Glycogenolysis, which is yung breakdown of glycogen to form glucose. Glycogenesis, formation of glycogen. And ang answer natin, which is glycolysis. So, yung process of glycolysis does not necessarily mean na yung pyruvate magiging siyang lactate. This only happens when it is an anaerobic glycolysis. In cases that there is the presence of oxygen or aerobic glycolysis, ang nagiging product natin instead of lactate is actually acetyl coenzyme A or acetyl CoA. So next question. What is the biomarker for bone resorption? The answer here is cross-linked C-telopeptides. Next question. What is the indication of relative concentration? This is an old recall question. The answer here is dilution. So, ito yung mga kailangan na natin i-memorize. Cross-linked C-telopeptide, bone resorption, relative concentration, dilution. So, ito. Next question. What is the indication of relative concentration? This is an old recall question. The answer here is dilution. So, ito yung mga kailangan na natin i-memorize. Cross-linked C-telopeptide, bone resorption, relative concentration, dilution. So, ito, which of the following tests uses p-nitrophenyl phosphate as a substrate? If you want more information regarding sa mga principles ng enzyme testing, then I suggest tignan nyo yung enzyme chapter ng book ni Dean Rodriguez. Pero in terms of yung gumagamit ng p-nitrophenyl phosphate as a substrate, the answer is ALP. Yung specific method na gumagamit ng p-nitrophenyl phosphate is yung Bowers-McComb or SAS modification of the Bowers-McComb method of ALP determination. So, try nyo rin aralin yung mga specific na tests for AST, ALT, ACP, at kung ano yung mga substrates nila. I know na very bulk itong part na to, pero marami-rami rin yung questions na lumitaw sa boards about enzymes. So, I suggest na aralin nyo talaga yung mga principles ng test nila. Next question is, which of the following is under the pre-analytical phase of testing? Common sense question. Pre-analytical phase, this occurs before the procedure or the actual method ng testing. So, if merong involved na instrument, automatic analytical phase na itong mga to. And if meron naman involved na results, automatic, tapos na yung post-analytical. Tapos na yung analytical phase, so post-analytical na siya. So, the answer is pre-analytical test order. Kasi ito yung only option that occurs before the methods or instrumentation is involved. So which of the following is indicator of poor nutritional status? So the answer here is prealbumin or transiteratin. So the rationale for this is actually lifted directly from HAR. So according to HAR, serum levels of transthyretin or prealbumin falls rapidly in patients with deficient protein nutrition. As such, prealbumin or transthyretin is used to detect malnutrition and to measure the patient's response to dietary supplementation. So indicator of poor nutritional status, transthyretin. Yun yung answer. Next question is how much diluent is needed to make 1:20 dilution with a 0.2 ml of serum. So, yung ginawa kong method dito is minultiply ko na lang tong 0.2 times 20. 0.2 times 20 is actually 4 ml. 0.2 ml times. So, ang 4 ml na to, ito yung overall volume. Ito yung total volume natin. So, if meron tayong total volume na 4 ml, and meron tayong serum na 0.2 ml, yung difference ng dalawa, that is the volume of the diluent na kailangan natin. So, the answer here is 3.8 ml. It's just 4 minus 0.2. 3.8. And the final question is, how is alcohol formed? Nagtaka rin din ako sa question na to, pero alcohol is usually formed from the fermentation of sugars. So, sugar, if it is fermented, will actually form alcohol. So that's it for our recall questions in clinical chemistry. So, before tayo mag-move on sa next subject natin, magbigay naman tayo ng konting tips on how you can survive the next board exams. Mag-take na muna tayo ng break from recalls and bibigyan ko kayo ng isang tip on how to survive the board exam. And isa tong tip na sinasabihan ko sa lahat ng kilala kong nagbo-boards, whether or not medtech ang subject. And ang tip na to is, do whatever it is that makes you comfortable. Now, maraming magbibigay sa'yo ng tips na, kesyo, kailangan 2 weeks before hindi ka na nagre-review, or 3 days before hindi ka nagre-review, or kailangan magpakapagod ka the day before para mahaba yung tulog mo ng gabi. And honestly, normal lang if hindi mo gagawin lahat ng yon. Okay lang, huwag kayong maging anxious na, oh my God, sabi ni ganito, sabi ni ganon. Gawin nyo lang kung saan kayo magiging comfortable. Kung hindi kayo mapakali, na hindi kayo nagre-review sa last week or sa last 2 weeks or sa last few days, or gusto nyo talagang mag-review kayo. Kasi same lang din. If kinakabahan kayo na hindi kayo nagre-review or anxious kayo, mas maganda nang mag-review na lang kayo. At least may na-accomplish kayo. Kasi ang point kung bakit hindi nagre-review ang sa mga last few days or sa mga last week ng boards is in order to relax their mind. And if hindi mo naman magagawa yun, okay lang. Kasi iba-iba tayo, guys. And you can do whatever it is that makes you comfortable and that you think can help you pass the boards. Kung saan kayo panatag, doon kayo. Yun yung aking tip to anyone that will take the boards or who wants to take the boards. Huwag kayong papa-influence sa ibang people or huwag yung i-overthink yung habits nyo kasi sinasabi sa inyo na ganito dapat, ganun dapat. Huwag kayong papa-influence. Basta kung saan kayo comfortable. So, now let's go back to our recall questions. Our next topic is, or our next subject rather, is microbiology and parasitology. Microbiology and parasitology. So, I can parang confidently say that around more or less 50-50 naghati yung microbiology and parasitology dito. Like, I do not recall if merong virology questions, if ganun karami. I think meron, pero hindi rin siya ganun karami. So, first question natin is, which of the following bacteria should be considered important when reading gram-stain smears of soft tissue abscess? So, sa mga ganitong questions, I usually just do a process of elimination. So, if Streptococcus pneumoniae, or Streptococcus pneumoniae rather, yung naalala mo lang dito is community-acquired bacterial pneumonia or otitis media and yung meningitis. So, I don't recall anything na meron siyang relation sa soft tissue abscess, so eliminated na agad. Neisseria gonorrhea, and soft tissue abscess, parang hindi familiar. And Pseudomonas aeruginosa, ito yung parang naging pinag-isipan ko rin, pero Pseudomonas aeruginosa kasi is usually associated with eye infections in contact lens wearers and yung swimmer's ear and yung mga nosocomial infections and also respiratory tract infections kapag a person is infected with cystic fibrosis, and parang hindi naman siya ganun ka-associated with abscesses. So, Pseudomonas aeruginosa, eliminated na agad. Best option talaga ito is yung Staphylococcus aureus. Kasi, I usually answer mo yung Staphylococcus aureus kapag related siya sa something na related sa skin, wound infections, carbuncles, ganun. Minsan mo lang may isip yung if may gastrointestinal infection, ganun. Pero Staphylococcus aureus, best talaga siya. Abscess, wound infections, and carbuncles, yan. So, it's the best answer. What is the best specimen for the isolation of Bordetella pertussis? So, Bordetella pertussis, as we all know, is the causative agent of whooping cough. So, lahat naman to, guys, they are respiratory tract specimens. But the answer here is nasopharyngeal swab or aspirates. Siya yung best na specimen for the isolation of Bordetella pertussis. Next question. The organism mainly responsible for the occurrence of otitis media can be identified by a positive result from which of the following tests? So, hindi directly asked kung ano yung organism na involved. Ang binigay sa atin is a disease. Otitis media. So, otitis media, related siya sa Streptococcus pneumoniae. So, how do you differentiate Streptococcus pneumoniae? Actually, there are a variety of tests na kakaiba yung result sa Streptococcus pneumoniae. Pero given these options, yung best answer dito is Optochin. Optochin, or Streptococcus pneumoniae, it is susceptible to Optochin, unlike other Streptococci. So, siya yung magiging differentiating factor niya. Positive ang Optochin susceptibility test niya. So, next question. Eto, maraming nainis dito kasi kalabas ko ng room ko sa board exams, meron na agad nagreklamo. Ang dami daw namin memorize na Invictus, at yung binigay nila is yung sa E. coli, which is yung pas-pas neg-neg. Pero huwag kayong papaloko guys. I swear, kailangan nyo i-memorize yung mga Invictus. Baka bigyan kayo ng mahirap. Kasi minsan, tititripan nilang magtanong ng mga hindi masyadong inuulit-ulit na bacteria. Pero sinwerte kami at binigay sa amin is E. coli. Next question. Hi, it's an image question. Buti na lang at madali lang yung tanong. Kasi yung tanong is, what type of biosafety cabinet is seen in the image below? So, I'm sure, or like, I'm very almost 100% sure na memorize nyo na yung mga biosafety cabinet. Kasi almost every subject, inuulit-ulit yung mga definitions nila. Ano yung difference nila sa isa't isa. Pero, parang iba na yung usapan kapag meron ng picture. Kasi kailangan mo na nun ng understanding. Hindi lang i-memorize mo kung ano yung word na nakalagay or like keywords. Kailangan naiintindihan mo talaga. So, syempre, nakikita naman natin sa biosafety cabinet na to. Ito yung picture na lumitaw nung boards namin. Ganito. Ganito siya halos. Nakikita naman natin dito na yung room air, siya yung tumatama sa specimen na nasa loob ng biosafety cabinet. Ay yung nafi-filter lang na air is yung lumalabas. So, yung na-protect lang is yung personnel. And yung mismong specimen, hindi siya na-protect. So, this is only associated with BSC1. I suggest if hindi nyo masyadong nai-imagine or nahihirapan kayong mag-imagine ng mag-analyze. I suggest na tignan nyo na yung mga pictures ngayon pa lang para at least ma-familiarize. Nyo. So, ito yung mga different types of I suggest if hindi nyo masyadong nai-imagine or nahihirapan kayong mag-imagine ng mag-analyze, I suggest na tignan nyo na yung mga pictures ngayon pa lang para at least ma-familiarize nyo. So, ito yung mga different types of biosafety cabinets. Next. Which of the following organisms is not caused by an obligate anaerobic bacteria? Which of the following diseases I think, which of the following diseases is not caused by an obligate anaerobic bacteria? So, parang tinatanong lang dito, yun nga, ano yung hindi obligate anaerobic bacteria? Very easy to siguro if linista nila yung actual name ng organism. So, unfortunately, hindi nila linista. Pero, if nag-aral kayo ng mabuti, memorize nyo na to syempre. Yung botulism, Clostridium botulinum, gas gangrene, Clostridium perfringens, tetany tetanus is Clostridium tetani. So, lahat ng to, they are under Clostridium. So, since under silang lahat ng Clostridium SPP, lahat sila is actually obligate anaerobic bacteria. Ang naging difference lang is sa tertiary syphilis, this is not obligate anaerobic. It is actually microaerophilic. Meaning, it can tolerate some amount of oxygen or it can survive at a small amounts of oxygen in the environment. Meanwhile, itong mga obligate anaerobic, they cannot thrive in an environment na meron oxygen at all. So, magandang trick din yun, like process of elimination na lang if alam mo kung ano yung specific na organism na nagkakos. Since pare-parehas lang Clostridium, yung kakaiba na lang, yung sa Treponema pallidum. Diba? Like, yun yung mga iba't ibang tricks sa boards. Next, how would you describe the species Actinomyces? I'm gonna be honest with you guys. Hindi ko to nasagot ng maayos ng boards ko kasi parang medyo na-blanco ako. Yung Actinomyces, it is a gram-positive bacilli that is facultative. Ito yung tamang sagot. This showed up 2 times in my board exams. Ang alam ko lang nun sa Actinomyces is that it was a bacilli, pero hindi ko maalala if gram-positive siya or gram-negative siya. So, dahil 2 times siyang lumitaw, magkaiba yung sinagot ko. dun sa 2 times na yon. And good trick naman kasi at least isa lang yung mali ko at yung isa naman tama. So, if hindi talaga kayo sure guys, yun na lang din yung tip ko. I know na parang medyo easy way, pero at least may tama kang isa, di ba? So, next question. Helicobacter pylori or H. pylori is a urease-positive organism that affects which of the following? So, the answer here, even if familiar ka lang sa nakakos ng Helicobacter pylori or hindi ka masyadong sure sa pathology niya or pathophysiology niya sa body, alam mo na Helicobacter pylori causes ulcers. So, what parang factor in the human body or like element in the human body can cause gastric ulcers? Syempre, an acidic pH of the gastrointestinal system. So, yung mechanism of action pala ng Helicobacter pylori is that it increases the pH within the stomach. Kasi ini-stimulate yung secretion of gastric acid or hydrochloric acid. So, yung hydrochloric acid na to, siya yung nakakakos ng gastric ulcers. Hence, Helicobacter pylori is associated with gastric ulcers. So, the uptake of free DNA by recipient cell is termed as, the answer here is transformation. Kasi free DNA. Dapat alam nyo rin yung meaning ng other choices. For example, conjugation, keyword dito, pili. And what is the form of DNA? Yung form ng DNA na nagtatransfer is in the form of plasmid. Pili, plasmid. Tapos sa transduction naman, it utilizes a bacteriophage. Ito naman, free DNA. Galing mismo sa environment. Now guys, alam ko na binigyan ko kayo ng keywords, pero in any case na hindi magiging direct to the point yung mga examiners and sasabihin nila yung definition ng plasmid and yung definition ng bacteriophage or baka yung definition ng pili, dapat alam niyo yung mga definitions nila. Like for example, yung bacteriophage, yun yung mga virus na nag-i-infect ng bacteria, yung plasmid, yung extrachromosomal DNA, kailangan familiar kayo doon kasi baka hindi nila ibigay yung exact word at hindi gumana yung keywords natin. Kailangan deep yung understanding nyo. Okay, so mag-move on tayo sa next. Next question. Egg yolk agar is classified under what type of medium? So, yung egg yolk agar, guys, it is a differential medium. So, before tayo mag-move on sa next question, try natin i-define to isa-isa. So, yung selective medium, guys, siya yung inhibitory talaga. Inhibitory except the organism of interest. While yung differential medium naman, guys, sila yung nagpapakita ng mga defining characteristics in order to differentiate. Exhibit defining characteristics. Defining characteristics. Enriched medium naman, sila yung ginagamit in order sa mga nutritionally demanding bacteria or yung tinatawag nating fastidious na organisms. Fastidious organisms. While yung enrichment naman, guys, wag nyo silang ililito sa enriched kasi yung enrichment, it supports some organisms but not others. So, yun yung mga general descriptions nila. So, in particular, yung egg yolk agar, ginagamit siya in order to detect lecithinase, lipase, and also protease activity. So, tandaan nyo yan, guys. Egg yolk agar is a differential agar. Next question. A profuse watery diarrhea, rice water stools. Yun na, nakita natin yung sagot. Rice water stools. Leading to dramatic fluid loss, severe dehydration, and hypotension that frequently leads to death is the hallmark of which toxin activity? Rice water stool. Inulit-ulit natin to. Rice water stool. Cholera. Rice water stool, cholera. Meron iba't ibang iba't ibang characteristics yung diarrhea, or if like meron sila ibang physical characteristics, possibly yung cellular content nila, na pwede nating ma-point out na, ito yung bacteria na nagkakos, ito yung toxin na nagkakos. Maganda na ma-memorize nyo yun. Like, yung difference, kunwari, if Shigella yung infection, or if cholera, ganun. Iba-iba yung characteristics na describe nila. Usually keywords. The keyword here is rice water. Rice water diarrhea is cholera toxin. So, what is responsible for the antimicrobial penicillin resistance of the Bacteroides fragilis? Keyword again, penicillin. Pag nakita mo yung penicillin, ano dapat unang pumapasok sa isip mo? Beta-lactam. Tandaan mo that penicillin is a beta-lactam antibiotic. So, even if hindi ko nabasahin tong Bacteroides fragilis, yung mind ko, automatic na siya sa beta-lactamase. And that is the correct answer. Beta-lactamase is the correct answer. Siya yung mechanism of resistance ng Bacteroides fragilis. So, which of the following is used for fungal cultures? So, the answer here is Sabrode dextrose agar. Iba-iba Tapos sheep blood agar, alam nyo to. Chocolate agar plate, cycloserines, cefoxitin fructose agar. Iba-iba sila ng purpose or iba't iba sila ng usage. Maganda if tignan nyo yung sa handouts nyo. Pero I also suggest na maging familiar kayo sa as many mediums as possible para alam nyo kung ano yung ginogrow nila specifically and if ano yung purpose nila. Okay, let's continue on to the next question. The next question is, what is the gold standard for SARS-CoV testing? Gold standard SARS-CoV testing. If you are familiar at all with the testing for COVID-19, you would know that the test that we use usually here for confirmation is RT-PCR. So RT-PCR is a type of nucleic acid test. So that's the answer. It's nucleic acid testing. At what temperature should clinical specimens suspected of containing viruses be kept for transport that takes days? So this question is lifted directly from Apollon. Apollon is another review booklet na may mga practice questions na pwede mong ring gamitin to review for the boards. Very relatively straightforward yung mga tanong dito. Kaya if gusto mong mag back to basics na questions, I'm very good na pang review yung Apollon. So yung question here is, kailangan mong tandaan is yung transport that takes days. Key term yung days dito, guys. And yung answer dito is 70 degrees Celsius. Yun yung answer sa Apollon. Pero, meron siyang question na directly above it na nagtatanong din ng temperature. And yung question naman dito is, specimens collected for virus isolation should be kept at. So, ito naman, yung key term ko dito is isolation, tas for virus isolation should be kept at. So, like, yung storage temperature niya. So, it's just 4 degrees Celsius. So, if yung transport nagtitake ng days, relatively mas malamig, negative 70 yun yung kailangan na temperature in order to remain viable yung ating virus. So, next question, mag-move na tayo sa mga parasitology questions. Trophozoite forms of amoeba is found in which type of stool specimens? So, the answer here is a watery specimen. So, depende sa consistency ng stool or kung ano yung makikita natin na forms ng amoeba. Since there are cyst forms, there are trophozoite forms. If formed yung stool, most likely cysts yung makikita natin dito. And mas more resistant din yung cysts sa environmental factors. Meanwhile, if semi-formed naman yung stool, makikita natin can be both cysts and trophozoites. And kapag watery yung stool, as I stated in our answer, makikita natin dito is yung mga trophozoites, like gumagalaw sila. They're motile. Next question, how do you differentiate Strongyloides stercoralis from hookworm larvae? Kasi hookworms, usually pa naka-identify natin dito is yung shell, yung eggs nila. Pero yung eggs nila, they have quite na thin shells. And may mga times na yung mga larvae, dahil nga thin yung shells, nag-escape sila from the eggs from the eggshell, and then pwede mong pagkamalan na strongyloides stercoralis. Pero there are differentiating factors dito. I'm sure na inulit-ulit na rin to sa inyo. Tandaan nyo na kapag strongyloides stercoralis, short buccal cavity, large genital primordium. Ito yung sagot. Short buccal cavity, large genital primordium. Meanwhile, yung hookworm naman is kabaliktaran, which is yung long buccal cavity and small genital primordium. Ito yung sa hookworm. Ayun yung strongyloides stercoralis, guys. Yung ginagawa kong palatandaan dito para hindi ko makalimutan. Strong siya. So large yung genitals niya. Like large, big balls. Strong ka, big yung genitals mo. Big yung balls mo. So, yun, kaya like kong tatandaan to. Strongyloides stercoralis, large genital primordium. Alam ko na nun na short yung isa, tapos small genital primordium, long yung isa, naman yung sa hookworm. So, yun lang yung kailangan yung tandaan. Guys, wag na kayo palang overthink or mag-guilty pa na baka treated SPG yung mga genomics or yung mga techniques nyo to memorize. Basta gamitin nyo lang yan if yun yung kailangan yung gawin para ma-memorize nyo. Hayaan nyo na. Mag-sorry na lang kayo after. Next question. In cysticercosis, humans serve as... this is quite a tricky question kasi sa cysticercosis, ito yung type ng infection caused by what parasite? Syempre, Taenia solium or pork tapeworm. So, yung pork tapeworm, like, more possibly, yung familiar na mode of transmission natin na naalala natin is, makukuha mo siya kapag nag-ingest ka ng undercooked pork. Kaya nga siya pork tapeworm. Pero, in these cases, na nag-ingest yung humans ng undercooked pork, hindi tayo magiging intermediate host. Magiging tayong definitive host. Kasi yung ini-ingest natin, in this case, is yung larvae na nasa flesh ng pork. Or nasa flesh ng pig. Yun yung ini-ingest natin nun. And nagiging silang adult sa intestines natin, tapos naglilay sila ng eggs, kaya may kita natin yung eggs sa stool ng human. Meanwhile, kapag naman yung other method ng pagkaroon ng infection with Taenia solium, ito naman yung mas malalang method or yung mas worse na disease, I can say, yung cysticercosis or yung times na ang humans nagsiserve as an intermediate host. Why? Kasi yung ini-ingest natin dito is yung egg ng Taenia solium. And kapag in-ingest natin yung egg ng Taenia solium, ang mangyayari sa atin is yung larval form na magpo-form from those eggs, sila yung makakapag-cause ng extraintestinal disease. Like, nagta-travel sila to other parts of the body outside of the intestines. So, pwede makapag-cause sila ng lesions in the cerebral cortex. Yung pagkakaroon ng lesions in the cerebral cortex, yun yung rin-refer natin as cysticercosis. So, in this case, yung in cysticercosis, yung humans nagsiserve siya as an intermediate host. So, huwag kayong paparito, guys. Kasi, pwede rin na yung itanong is, in Taenia solium infections, humans serve as, kapag ganun yung tanong, syempre, both definitive hosts and intermediate hosts, like A and B, ganun yung isasagot nyo. Pero kapag cysticercosis lang, intermediate host lang. Kapag naman nag-ingest lang sila ng, kunwari, problem sa intestinal infection, tinea solium, intestinal infection, definitive host. Dahil nag-ingest ng undercooked pork. Ganun lang yan. So, dahil sa cirrhosis yung tanong, yung sagot natin is intermediate host. Next question. Which organism is indicated when disease is caused by humans ingesting an egg with an oncosphere? I think sinagot ko na to sa past question. Ang answer dito is Taenia solium. Kasi yung Taenia saginata, Taenia asiatica, and Diphyllobothrium latum or Dibotriocephalus latus, yung new name. Etong mga to, hindi mo sila ma-acquire. Hindi kayo maka-acquire ng disease dito if mag-i-ingest lang kayo ng egg. Ang ini-ingest dito is yung actual flesh of the animal involved. For example, sa saginata, kailangan beef, undercooked beef. Sa asiatica, undercooked pork din, like solium. And sa Diphyllobothrium latum, it's undercooked fish. Yung larval forms within their flesh yung ma-ingest natin in order to develop infection or develop a disease. Meanwhile, in Taenia solium, since there are two forms, isa na in-ingest natin yung larval form through undercooked pork and meron din yung na-ingest natin yung egg niya na nagko-contain ng oncosphere, then siya yung tamang sagot. The whip-like motility of paramecium is attributed to its, the answer here is cilia. Guys, huwag kayong papalito. Baka pag nabasa nyo yung whip-like, isipin nyo agad, whip-like, flagella. No, it's cilia. Paramecium has a cilia.